Right, so this is still going through that overview of the immune system. And just to take a step back and look at what we have seen briefly before and what we're going to focus on in this lecture. Right, so the learning objectives for the first week, I know that it does look like a lot, and it looks like a whole course of immunology in like 2 lectures, right? So there is kind of like impossible. It does feel like drinking from the fire hydrant, right? So I try to minimize that feeling, I would say, down to a fire hose, right? Instead of maybe a garden hose, not a fire hydrant kind of thing, right? So what we have looked last time was basically on the subdivisions of the immune system, and we are looking at all different levels of protection and all different types of strategies that the immune system has to defend you against pathogens and also against your own cells that go crazy. Okay, so we looked at first, and we're still going to look more into this first line of defense, which is really all part of what you are born with, right? So first line is really relevant in relation to physical barriers. So you do have your skin, you have all the mucus on the organs that are in direct contact with the external world, right? So you have mucus that are part of your GI system that goes all the way from the mouth to the anus. You have mucus in your respiratory system and also genitourinary system, right? Besides skin and mucus, you also have your friendly bacteria, so those are your commensals. And mainly you're going to find the commensals when you talk about commensals, you're really talking about bacteria that are present in the intestines. But keep in mind that commensals are also bacteria that are present like everywhere else, and that includes on the surface of your skin and all other mucous membranes. Besides that, you also have chemical barriers, like for example, the pH in your stomach, right? Being that pH is very acidic in the stomach, meaning that if you swallow some of these pathogens, most likely they would not survive the acidity of the stomach. And then besides, the chemical barriers, you also have enzymes that their jobs, they're really to break down components of pathogens in order to, digest them or make them ineffective or inactive. So this is really the first barriers, the first lines of defense. And then we're looking at differences between what you're born with in comparison to the system that you need to develop, right? So you are born with all these physical barriers and enzymes and chemical barriers and cells that are always in a certain way. And they do not get modified. They do not retain memory of what they have seen before. So those are all part of your innate immune system. Okay. So now we're going to look a little bit more into the differences, right, between innate and adaptive immune system. And we are going to also start looking at the organs that are responsible for the development of the immune system. So we're going to be looking at the primary organs and at the secondary organs. At least we're going to see what they are. Okay. Besides that, we are going to start looking at how is it that cells recognize things, right? So briefly going to take a look at pattern recognition receptors. And as I mentioned before, these recognition receptors, they are important to detect patterns on pathogens, but they are also important to detect patterns in damaged cells. So your own damaged cells. Okay. Then we are still going to take a look at strategies of getting rid of bad cells, right? So basically how the immune system kills things. And one main action, main mode that these cells have to kill foreign things is really to eat them and digest them. So that is phagocytosis. Besides that, you can also have specialized molecules that are injected into pathogens. So that is called pathogen-directed killing. And this is important, especially when the pathogen is bigger than the cell. Okay. Besides that, you know that you have intracellular pathogens, and you cannot really, get this cell to or get some of those pathogens killed. So you end up having to kill the entire infected cell in order to kill the pathogen with it. Okay, within it. Okay. So after all this, we're also going to look into how cells of the immune system communicate with each other. Mainly, they are going to rely on receptors on their surface. Okay, so the receptors have to be activated, and they can either be activated by molecules that are released in the environment, or they can be activated by cell-to-cell contact, these receptors. Those are the two modes that immune system cells communicate. Okay. Then after all this, we will start to take a look also at how you slow down and stop an immune response. Because if you don't, then you're going to have a problem. If you're not able to eliminate the action of all of these immune cells, they will cause chronic inflammation, or they will start attacking your own cells, and that's very much detrimental. Okay. All right, so we had stopped at this slide in the last lecture, and I had mentioned that this inborn function or inborn characteristic of the immune system has to do with not being susceptible to infection by certain pathogens. In this example was if you don't have the receptor for a certain virus, in this case HIV, then you will not allow your cells to be affected by the virus. Because when viruses infect cell, each virus has a certain tropism, meaning that each virus has a certain receptor or a certain characteristic in the cell membrane that they need to bind to in order to infect that cell. If the cell does not have the receptor, then the cell cannot be infected by the virus. Okay, and we see examples of like the cold virus. Right, we have influenza virus that they go through a certain receptor on cells that are in your respiratory system. Right, you have examples of coronavirus that infect not only cells of the respiratory system, but they start infecting cells of your GI tract. Right, so if you have the receptor, if the cell has the receptor, it is susceptible for viral infection. So in this case here, what was done was that one of the co-receptors for the virus, for the HIV virus, one of these receptors was removed from the genome of those embryos. So this, this was before fertilization. So say it was removed from the ovum, right, of those of the embryo. So after fertilization and after the babies were born, basically they do not have those receptors. That means if they are exposed to HIV, which actually would be the case because the father has HIV, right? So if the babies, when the babies are exposed, they would not really be infected because they don't have the receptors for that. And this whole idea of not having the receptor, you should also raise a question in your mind, saying that is it possible that some people naturally don't have the receptor? What do you think?
Yes.
Yes. It's possible. Some people don't have the receptor, so are they infected by HIV? No, right? So it's a good thing, right? But the percentage of the population, I don't really know what's the percentage. And it could also happen that the person, although the person is not infected by HIV, the person could carry the virus to, someone else, right? So it still could infect somebody else, and the person is not really infected. Don't ask me mechanisms, like use your imagination. Right? All right, so here is from the book showing the different levels of protection, right? So we really start with the physical and chemical barriers, right? So we start here with your innate protection. Then after this, if your pathogen is able to get deeper, right, to pass the initial barriers, then it automatically is going to find a lot of proteins that are floating around in the interstitial fluid, and they are floating around in the blood and in the lymph. Okay, so those proteins are mainly produced by the liver. Okay, when you talk about complement proteins. Okay. And besides that, you also have certain proteins that are naturally antimicrobial, okay? So you have proteins that will cause some damage in the pathogen. But here, what I like to talk here is really about the complement system. Okay, so these are produced by the liver, and they're always floating around, but they're really floating around in an inactivated form. If they find a pathogen, then they become activated. Okay, and you have, as we're going to see next week, you have like 3 different pathways for this complement to become activated. Okay, so this is really an interface between the innate immunity and the adaptive immunity. Is really like complementing both types of immunities. Okay. Now let's say that the microbe did not get damaged by complement system. Now you're going to have your innate immune cells that jump into action. Okay. So those are going to be your granulocytes and your agranulocytes, also your natural killer cells, okay, and some cells that like to sit around in the epithelial tissues, right? Those are more like macrophages. They also called Langerhans, or I forgot the other names, mainly macrophages, right? So innate immune cells are going to jump into action. Usually the innate immune cells are very efficient. They can eliminate a lot of the pathogens that you're exposed to. The issue with this is that we don't really know how efficient they are, because if they are eliminating the pathogen, then you're not really going to get sick. So we can't really study something if you don't have, some disease to look at, right? So they eliminated the threat and you're still healthy. So how efficient they are, I don't know, right? So it's kind of difficult to say how efficient they are. But they're pretty efficient. Okay. Now, let's say that the pathogen was not killed by your innate cells. Now, at the same time that your innate cells are killing your pathogen, they are also communicating with the adaptive immune cells. So they are killing the pathogens, and they are also telling your adaptive immunity, it's like, look, there is a pathogen out here. Hopefully I'll be able to take care of it. But if I am not able to take care of it, be aware that we might have to mobilize the 3rd line of defense. Okay. So if those guys take care of the problem, then your adaptive system will never have to jump into action, but they're already primed with the presence of that pathogen. Now let's say that these cells did not take care of the pathogen. Now your adaptive immune cells will have to jump into action. And you're going to have to activate the B cells and the T cells, and you're going to start producing antibodies, and you're going to, be really like have a lot more of tools to eliminate the pathogen. Okay, so all these different levels of defense. I do have better slides for that. Okay, so here to really reiterate the whole concept of innate immunity, right? So the first line of defense. And one thing that we say is that innate immunity is not keep a memory, it's not going to remember what it's seen before. So it's going to react always the same way. It doesn't matter what pathogen it sees, okay? Now, reality of this is not exactly like that, because there is some level of memory, but that is like beyond what we have to study for this basic class, but so it's like but there is a little bit of memory. You're going to see that if you go to medical school or to other to graduate programs related to immune system function. So basically, for all that we are concerned, innate immunity does not have a memory, and they also work in a non-specific way. Means that it doesn't matter what they see, they're going to react the same way. Okay. So your innate immunity, as we mentioned, involves the physical and chemical barriers, and the cells of this system, they have the receptors that are coded in the DNA, and they do not get modified. They're always the same receptors. What are those receptors? Those are your pattern recognition receptors, okay? Pattern recognition molecules. Besides the receptors on the surface of your innate cell, you also have those proteins that are floating around that are part of the complement system, right? So complement system, most of those, they're really part of innate system. Okay. All right, so here are some examples of chemical and physical barriers, right? So make sure you just go over that, like I mentioned. Okay. All right. So in comparison here to your adaptive system, you are not born with the receptors that are on the surface of your adaptive cells. Okay. When you talk adaptive cells, you're going to talk about B cells and T cells. That's it. So there are lymphocytes, B cells and T cells. You also have other lymphocytes like natural killer cells, right? But the natural killer cells, they are part of innate system, not of adaptive system. So for adaptive is only B and the T cell. So those are the ones that they are going to modify their receptor in order to identify on specific pathogen. So they are really, really specific for the pathogen. Okay. So after they modify the receptor, they are always going to keep a memory or save some cells as memory cells that if you find the same pathogen again, then you'll be able to quickly activate your adaptive response and eliminate the pathogen. Okay. So important thing about this, they do have memory and they are very, very specific. So since you are not born with it, in order for you to develop that receptor and in order for you to develop the memory, you have to be primed with the pathogen. Okay, so first exposure is always necessary for the adaptive immune system to develop. Now, this is actually one disadvantage because when you are first exposed to the pathogen, it takes at least 10 days for these cells to develop and for them to start mounting this adaptive immune response. Okay, so this is a disadvantage. It takes some time. The advantage of the adaptive system is that it will keep a memory of what it has seen. Okay, so second time that you're exposed, then you're going to have this big reaction. Now, this here, let's say this is a B cell that is a memory cell. So it had seen the pathogen before. It has a specific receptor for that mean bacterium, right? So now when it felt the presence of this guy a second time, what it happens is that it activates itself to start dividing a lot. Okay, so this one cell that recognizes the pathogen, what it does is that it undergoes this clonal expansion. That means it's one clone that has to do with the production of this receptor, that this clone keeps on dividing and dividing and dividing, so that you're going to have a whole army of cells that can recognize this type of pathogen. All right, so this is what is meant by clonal expansion. Okay, so after you kill the pathogen, there is no more pathogen to deal with, and then you're left with this whole army here looking for pathogen, and the cells are kind of bored, and without a job to do, they kind of die. They die out. So when they start to die out, this is what you call clonal deletion. Okay? So in the presence of the pathogen, you're going to have continuous stimulation, you're going to have the clonal expansion. When the pathogen disappears, then you're going to have clonal deletion. But you're always going to keep a few memory cells just in case you find that pathogen again. Okay? Okay. All right, so here are some very good properties of your adaptive immune system. So I put it here just to have it, summarized for you to study. Okay. All right, so here's some more comparison between your adaptive and innate immune system. Remember, innate you're born with, and the stuff is already there in the cells, already in the tissues. And the major player here are really your macrophages. I mean, your phagocytic cells, right? So you have macrophages and you have neutrophils. Neutrophils are important for surveilling bacteria in the blood. Macrophages are important to surveilling stuff in the tissues. Okay? Comparison with your adaptive system. Remember, you have to have priming. You have to have specific recognition by a receptor, and the cell needs to be activated. You need to undergo clonal expansion, and that is not going to happen just anywhere. It has to happen in specific meeting place for these cells. So this is when you get the involvement of secondary lymphoid tissues. So the recognition of the pathogen and the expansion of adaptive cells is really going to happen in secondary lymphoid tissues. Okay, so that is, those are lymph nodes, and also in the spleen, and also in gut-associated type of secondary tissues, like gout, mouth, and all this other fun stuff. So here a bit more on charts on comparing the two systems, and a bit more here too. I like this little table here because it tells you the cells that are involved here. Okay, so innate cells, you have those white blood cells that are mainly from the myeloid lineage, right, which you're going to see also next time. But also you have your natural killer cells here, okay? Natural killer cells are lymphoid lineage. These are the only ones that are part of your innate system. Now when you go adaptive system, you're going to have B cells and T cells, and you're also going to have your antibodies. Those are soluble proteins that are also considered as your adaptive system because they're very specific. Okay. All right, so I mentioned this one before. I think this is a duplicate slide. All right. So here is a comparison of how quickly those systems will jump into action, right? So you have here your innate system that jumps right away into action, and it lasts for days. Now, when you compare it to initial activation of your adaptive system, you know that it takes time, right? And in order to develop memory, it's going to take weeks or at least 10 days, depending on the pathogen, okay? Once you develop memory, a lot of times you have memory like for the rest of your life, right? Okay. So this slide here is really like the overview of the whole immune system, okay? So one thing to keep in mind is that innate and adaptive systems, they're really working together. Okay, they have to work with each other. Oh, come on in, come on in. Wrong class, don't come in. Well, okay, now let's get somebody else for immunology. Okay, so, all right, so what happens here is that they have to work together. They have to cooperate with each other, okay? The way they're going to work together, they are going to send signals to each other. They're going to send cytokines and chemokines, and they're going to interact with the receptors. They're going to bump into each other and pass some important information, right? And overall, once your innate system jumps into action past the cellular level of attack, then your adaptive system is going to be informed of this process of what they're fighting against, okay? So first thing here, you pass through the barrier, you have your innate cells that are going to follow the breadcrumbs of cytokines and inflammatory cytokines, and they do the chemotaxis, right, to get in there. And besides that, you also have the resident cells here. Like you see here, this star-looking cell. So this is a dendritic cell that catches the pathogen, and then it gets into the lymphatic system and travels all the way to a meeting place. So let's say this is a lymph node, right? It travels to the lymph node, and in the lymph node, this cell is going to look for a T cell, and is going to look for a matching T cell that can recognize the pathogen and is going to activate the T cell. So this is the first level here that you're going to see a clonal expansion of your T cells. So you're going to have T helper cells, T cytotoxic cells, and then the helper cells are going to interact with specific B cells. So those are your lymphocytes here getting activated, and you're going to undergo clonal expansion. You're going to start producing antibodies. Then those cells are going to get out of their home here on the secondary immune organ, and they're going to get into the circulation, and they're going to travel to the site of infection in order to fight the pathogen. Okay? Then after the pathogen is killed, then you're always left with some memory cells. Okay? Now, another important thing in this slide here is that it is showing you a bone. In this case here, is showing you a long bone. I would say that's the femur, right? And here you're going to have this in case of growing children, right? You can have red bone marrow in the marrow of the femur. But as adults, we really have the red bone marrow in flat bones. Okay, so it's not really in the long bones. So here, you're going to have the bone marrow here. In the bone marrow, you're going to have development of all of your blood cells. So we're talking about innate cells, all of those granulocytes, subgranulocytes, all the innate cells. You're also going to have development of your red blood cells over there. Okay, and you're going to have the development of your lymphocytes. So that is your B cell and your T cell. Now the B cell is going to develop fully in the bone marrow. That's why it's called a B cell. Okay, it fully develops there. By the time it leaves the bone marrow, it is mature. Okay, it is practically mature, ready for action. So all the development of this of the cells is in the bone marrow. So the bone marrow is a primary lymphoid tissue. Okay. Now the T cells, they come out of the bone marrow and they migrate to the thymus. In the thymus, they finish up their development. So they're still developing and they do not come out. They're only going to come out of the thymus when they're fully developed. So the thymus is also considered a primary lymphoid organ. Okay, so these two things here, those are your two primary lymphoid organs. bone marrow, and the thymus. That's it. Everything else that's related to lymphocyte development and meeting and everything else, those are secondary lymphoid tissues. That includes the lymph nodes and the spleen and the gut-associated nodes or places. Sorry? The last one, gut-associated? Yeah, the gout, gut-associated lymphoid tissue. Yeah. Okay. So here is just a brief introduction to the major cells, right? So you have here your innate immune cells. The dendritic cell is part of your innate cells, but the dendritic cell is between the innate and the adaptive, right? So for your innate cells, you're going to have here your neutrophil, eosinophil, basophil, monocyte, all right? And then the dendritic cell is going to catch the pathogen and is going to present it to the T cell mainly. It's going to be cross-linking the innate and the adaptive response. Okay. So here, this is a very classic type of graph that shows the typical response of your adaptive immunity. Okay, so let's see what is happening here. At first, you are going to expose your body to an antigen. So this day 0 here is your priming day. You're going to be first exposed. You're primed. Okay, so what's going to happen? First thing that's going to happen, you are going to get your innate immune system cells to respond. So here is your response of your innate immune system. Now, after the pathogen gets eliminated, if you get exposed again to that pathogen, here is your response of your innate immune system again. Exactly the same response as you had before. Okay? It's going to always respond the same way. Now here, in comparison to what happens to the response of your secondary or of your adaptive immune system, this is what's happening. At first, your adaptive immune system gets activated with the priming, but nothing is happening. Nothing is happening until about 10 days or so after first exposure. Then you have this big response of your innate immune system until the pathogen gets eliminated. Okay? So big response after about 10 days. Now, second time you're exposed to the same antigen, so antigen A, look at the response of your adaptive immune system. It's right away and is huge. It's a lot bigger than your initial response. So when you see this very much amplified response, you know this is a secondary type of response. Okay, so let's say now that you're going to get exposed a third time to this antigen, you're also going to have a secondary type of response. It's not like the third time you're going to get even more response. No, you're going to have this amplified secondary type of response. Okay. Now, what happens here, if you put in another antigen, like for example here, if you start here, I have it on mute. But of course, my husband has to call when I'm teaching. It happens every time. Sorry about that. Okay. So, what was I saying? Antigen B. Oh, there we go. Here. Let's put antigen B right here. Okay? How do you think your innate immune system is going to answer, is going to respond to antigen B? Exactly the same way. So here is a response of your innate immune system to antigen B. And how is your adaptive immune system going to respond to antigen B? Same way as what? As antigen A when it was exposed the first time. Because now this is the first time that you're exposing antigen B, so now the response for the adaptive is going to be like this. Quiet, 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 and a primary response right there. Okay? So for antigen B, you're going to have a primary response. Okay. Now, if after 30 days or so you expose antigen B again, then you're going to get your secondary response to antigen B. Okay. All right. So everybody's cool between differences of primary and secondary response? Yeah. I'm going to ask you something about this.
Yes.
Yes. Yes. Everybody's awake? Yes. Yeah. Okay. So here we get to talk a little bit about the types of types of immunity that you develop, right? So look at this. Immunity in general, right? You have adaptive or innate. Innate is always the same, no memory, right? Now considering the adaptive immunity, now you have this branching business going on here. So how do you get adaptive immunity? You can get it naturally, or you can get it artificially. Okay, so let's look at the natural type of defense, an adaptive immunity first. You can get it passively, or you can get it actively. So when you're talking about a passive, natural, adaptive immunity, first of all, when you're talking about adaptive immunity, you are talking about antibodies, and you are talking about B cells and T cells. Okay? So either antibodies or B cells, specific B cells or specific T cells, all right? So let's say that you are getting some adaptive immunity naturally. Now, if you're not developing the disease, that means you are receiving antibodies from somebody, okay? So if it is natural way to receive antibodies from somebody, the only way that that can happen is via breastfeeding, okay? So natural passive way of getting adaptive immunity is by receiving antibodies through breast milk. That's it, okay? Now another natural way to develop your own B and T cells to be specific to a certain pathogen is by getting infected with that pathogen. Okay, so let's say that you were a baby and then you get sick with a cold, so that's a rhinovirus. So now you develop your own cells, memory cells against rhinovirus. So there was an active, natural type of infection. Yeah.
Let's say the mother is breastfeeding and she gets like the flu vaccine. Will that like?
Okay, if the mother is breastfeeding and she got the flu vaccine. So the mother is going to develop cells that are going to produce antibodies against the flu, but those antibodies are mainly in the blood stream. I'm not sure if those antibodies are going to cross. Yeah, that's a more complicated question that I was expecting. If the antibodies cross to mucous membranes, or if the antibodies are able to cross through depending on the type of the antibody, they might be given to the baby or not. So we're going to look into that when we get to antibody types and functions, okay? So if the antibody is exported through if the antibody is capable of going through the breast milk, then the baby will get it, but depending on the type of the antibody, the baby might not get it, okay? But you getting vaccinated is not going to affect the baby really. You might give him some antibodies, might not.
Yeah.
Okay? Okay, so here. So natural way of getting secondary, getting adaptive immunity is, either with the antibodies from mom or by getting the virus and developing the antibodies yourself. Okay? And here comes the other way of getting adaptive immunity, artificial ways. Okay? The most common artificial way of getting adaptive immunity is by vaccination. Okay, so active artificial means immunization or vaccination. Okay? Now, an artificial and passive way of getting antibodies is by getting antibodies being transferred from some other animals that made it, but you are not drinking this through breast milk, you're just getting it straight into your veins. Okay, so that's artificial. Okay, so you can get passive antibody transfer straight into your blood. That's artificial secondary immunity. Okay. All right. It's going to get easier when we get to those parts later on. Okay. So here is just a word explanation of what I just said. Okay. And here finally comes your first iClicker question. Now I don't remember if I put this to count up or count down. Most likely it's going to be count down for 30 seconds. So let's examine the question and know the answer so you can get all your points, right? COVID. Yeah, you should be able to use that. This is on. I did not open yet, okay? So make sure you have the right answer. Although there's only a 50% chance of getting it wrong now, right? Only two choices. Okay. COVID antibody serum therapy. What is this? COVID antibody serum. That means when COVID was really, killing a lot of people, then there were some people that survived COVID, so they donated the blood. They had blood separation, isolation of the serum. So the serum had the antibodies. Now you're saving lives of people who are getting infected with COVID by doing the transfusion of those antibodies, just putting the serum into somebody's vein, okay? So this is what type of adaptive immunity? Is it passive or is it active? Okay, so now I'm going to open here and let's see. Okay, I got this, I got select. So yeah, so now it is counting and now I can see people voting. Okay, 8, 9, okay, so it's counting up. I don't think you need more than 30 seconds, but this is the time that cheating is allowed. So, you can talk to your neighbor and make sure you have the correct answers.
It says received.
Okay. So here is telling me that I have 25 responses. Does anyone need more time or any trouble with the iClicker? No, everybody's good? Okay, so I'm going to stop it. What is your answer? A, it is passive. Okay. All right. Okay, so this is another little summary of what I've been talking about. I'm going to skip through this because I want to get to the parts that are a bit more challenging. Okay. So here, different levels of immunity. Remember, you start with your innate immunity, you start with your physical and mucus secretions, then you go through your innate cells, and then you start here with your adaptive immunity, right? So innate is really fast, adaptive takes its time, and then it has its advantages, right? So when you look at adaptive immunity, you still have subdivisions of adaptive immunity. You can have the stuff that is floating around in the blood, mainly antibodies. So that you call humoral immunity. It's not like it's funny, right? But actually, the way it got its name was because in the ancient times, people used to say that the liquids in the body were called the humors, right? So proteins floating in the blood, that was called humoral immunity. Okay, so this really has to do with antibodies. Okay. Besides the antibodies and floating proteins here, you have your cells of your adaptive immune system. So that is cell-mediated immunity. Okay, this is when cells interact with other cells to cause some type of elimination of pathogen. Okay. Then when you talk about adaptive immunity, there is cell-mediated. This is mainly relevant to T cells. Okay, so humoral immunity has to do with B cells that produce antibodies, and then cell-mediated immunity has to do with the T cells. They're doing cell-to-cell combat. Okay. So here just to show you a better illustration of passive barriers, right? So all the organs that are in contact with the outside, they have this lining of mucus, and and here you have your, mucous membranes. And basically, in the mucous environment, you have its own, specialized trapping of pathogens and enzymes and ways to get rid of the pathogen. Okay. Okay. So here we start getting a bit more on strategies for killing the pathogens, right? And we are going to look into a lot more detail on those strategies. So here I take a step back and we're going to talk a little bit more about the artificial active form of immunization, right? So basically when you start talking about vaccination, right? Because when you prime your immune system, you can either prime it with the natural pathogen, or you can prime it with a not so pathogenic version of your pathogen, right? Or with what you call it attenuated version, or not so bad, right, of the pathogen that can still teach your immune system to fight the original thing, but at the same time, it shouldn't kill you. Okay, so what happens here? In case of smallpox, okay, in case of a smallpox type of infection, did anybody hear about the story on how the vaccination against smallpox happened? No? So this was Jenner, right? There was this scientist, physician scientist, back in, I think it was late 1700s, right? I'm very bad with history, right? I think I was always studying biology in my history classes. Don't do the same, I nearly failed. Okay, so what happens? What happens here is that there was this, a lot of problems happening with smallpox that is actually quite deadly, right? And then Jenner realized that there was a certain population of people, in particularly some women, that did not catch smallpox. Okay, so he started to study and started to do some associations that these women that did not catch smallpox, they were actually the women that were milking the cows. You know what I'm talking about? Like old style, milking the cows. They go like a cow and get milking the cows, right? And then Jenner realized that the cows had a similar disease to the human smallpox. And at the same time, the cows were kind of like, okay, not quite dying. Some cows died, some other cows didn't die. But then he realized that the women that were milking the cows did not get smallpox. So he was thinking that these women were probably developing immunity against the smallpox and not really developing the disease. So what he did was that he decided to test out his hypothesis using a small child, an 8-year-old child, an 8-year-old boy. And the story of that boy was that the parents had already lost, I don't even know how many children, to smallpox, right? And they were terrified that this boy was also going to die because of smallpox. So this guy, Jenner, went there and he said, you know what, let me get like the pus of one of those, pustules of the cow, and I'm gonna expose the boy to that, whatever is in that cow pox thing, and let's see what happens, right? So he exposed, the boy to whatever was in the cow pox thing and waited for some time. And then he exposed the boy to the virus, to the smallpox. And sure enough, the boy did not die. Well, if the boy would have died, we would not ever have heard about this, right? And considering that this was 1700s or something, I mean, talk about IRBs, right? And this was an innocent boy who may not have given permission to serve as a human experimental subject. A very risky experiment with an N of 1, right? So the boy survived. And this is how the idea of vaccination really got incorporated into a modern way of looking at things. Was it the first time that it was done? No. Ancient Chinese medicine was using this type of inoculation way before. But with the modern textbooks, we only hear about Jenner, right? He officially put in the publication, let's say. All right. So then, after the development of this, after the usage of the cowpox as a vaccine for smallpox, then a lot of other vaccines started to get developed, right? So here we have examples of vaccines that were very successful. So for example, here you have the total eradication of smallpox around 1977, right? And this guy here was actually the last human case of smallpox, and he had survived. Now, just because the disease was eradicated, it doesn't mean that the smallpox virus was totally eliminated from the face of the planet. That's not the case. There are a couple of laboratories that have the virus frozen. And you want to guess which two laboratories have it?
CDC.
Yeah, CDC and Russia. Yeah. How do you know about Russia? Oh, you did virology before? Oh, okay, yeah. So what are you doing here? See, don't answer. Okay, CDC and Russia. Yeah, in Moscow they do have this virus in the labs. It's frozen, right? It's kind of scary. Anyway, I don't even know if I should be more scared of the CDC or Russia. I'm not saying that. Okay. So Louis Pasteur, what did he do, right? So Louis Pasteur, what did he do? He realized that not only different species of viruses, like cowpox in comparison to smallpox, would be effective for bringing on immunity, right? So for adaptive immunity, but he also realized that some bacteria that were causing disease in the same species, those could also be modified so that they would not cause disease in the same species and would provide protection. So what did he do here? He got this cholera bacteria, and it's called fowl cholera, and fowl has to do with birds, okay? So this bacteria does not cause disease in humans, but it causes a lot of disease in birds, okay? It's avian type of cholera, okay? So if you have the fresh bacteria and you infect a chicken, so the chicken would die. Now what he did is that he started to culture the bacteria, and then he used this old culture of bacteria and inoculated fresh chickens, and then the chickens did not die. And then he got the fresh bacteria and inoculated the chicken that had gotten the old bacteria, that was vaccinated by the old culture, and he found that those chickens, they had protection against the fresh fowl cholera. Okay, so this was a way that he figured out that cultured bugs would be attenuated or would be less in pathogenic effect, and could be used as vaccines. So he called this whole process vaccine in honor of Jenner, that had used the cowpox, and vaccine or vaca, it comes from cows, right? So this is how he called the vaccination procedure. Okay? So these old cultures are live bacteria, but they're just not causing disease in that same species. All right. So vaccinations, they are still going on, and it's very, very important to get vaccinated, because if you expose a population to the wild type virus, a lot of people, they're going to die. Okay. Now the whole idea of getting populations vaccinated is that you can get enough people to develop secondary immunity, mainly herd immunity, And this is really, really important because there are certain people that have problems with their immune system, or they fall into these categories of vulnerable populations, like very young children or older adults, elderly, that are with their immune systems that start to go down. So if you have most of the population that are immune to a certain pathogen, then you can really keep that pathogen out from the entire population, and this herd immunity can really prevent the vulnerable subjects or the vulnerable individuals from getting the disease. Okay, but in order for this herd immunity to be effective, it really depends on the pathogenicity of the virus, and it depends on the total number of people that develop immunity. So as I say, for example, for the COVID virus, I think the number would be close to 80% of the population. So if you reach 80% of people that are immunized, then you have reached a herd immunity. If you have about 70 or 50% of the population, then you will not be as effective, and you will not really be protecting the people who are more vulnerable. Okay, and here comes another iClicker question. So since it is counting up, I'm just going to open it. Okay, so the question here is on the definition of herd immunity, and the question was also in the previous PowerPoint, so I'm assuming that everybody has read it. Everybody's like really quiet. I think I'm missing a couple of people. Okay, just put in somewhere. Okay, 25 answers. What is your answer? It is D. All right, so other things other than getting your own adaptive and innate immune system that really contribute to not getting sick are some things that to us they're really obvious, but there are some populations or some areas in the world that don't really have those factors, right? So those are clean water and proper hygiene and proper food, although I'm not really sure that we actually fall into the proper food category in the world. Well, if you try to eat whole grains and fruits and vegetables, not a lot of pesticides, then you're eating properly, right? Because your food is really going to develop your immune system cells, right? And your immune system in general, okay? Okay. Other than that, you also have options of antibiotics, and that could be either regular antibiotics that are against bacteria, or you could have antiviral drugs that are also effective against certain types of viruses. Okay. So here we start talking a bit more on this idea of antibodies, right? As we mentioned before, antibodies have to do with B cells, right? So B cells are going to produce antibodies, and the antibodies are going to be floating in the humors. So those are very important for your humoral immunity, right? So other than your antibodies, you also have certain proteins that are floating in the humors, and those are part of your complement system proteins. Okay, so humoral immunity really has to do with proteins floating around in the fluids. Okay, so those are part of your humoral, and in comparison here to your cell-mediated immunity, so this has to do with the function of your T cells. Okay. So here, just to break side by side your adaptive immunity, this is your humoral, producing antibodies, and this is your cell-to-cell contact with the two T cells. and these are cytokines, okay? So T cells are going to have to do contact, direct contact with other cells, other types of T cells. All right. So here's another slide with they're showing you the idea of clonal selection, okay? And we are going to talk a lot about selection of non-self-reacting lymphocytes, okay? So selection of non-self-reacting lymphocytes has to do with, now the word came out of my mind. It will come back in a minute. Now the chiaia bruto, okay. All right, so emphasizing here also your primary and your secondary lymphoid tissues. Remember, your primary has to do with your bone marrow and your thymus. Thymus is a gland that is right in front of your heart, close to the aorta, okay? So thymus and your bone marrow, those are primary. Everything else is secondary, okay? All right, so here is on that idea of how you recognize patterns, right? How your innate cells recognize patterns. They have those pattern recognition receptors, and they're going to recognize things that are on pathogens, and then they're also going to recognize things that are on damaged cells, patterns of damaged cells. Okay? So this part on pattern recognition receptor is really going to show up in chapter 3, okay? But I wanted you to start getting an idea of what is happening here, because it's going to be very interesting how you distinguish the activation of these cells, okay? So what happens here is that you are going to have on the surface of pathogens, in this case here is bacterium, you're going to have certain molecules that show up, and they can either be peptidoglycans, or they can be made of peptides and sugars, or they can also be made of some lipids, okay? But the idea is that they show up a lot on the surface of pathogens, but they're not really showing up on the surface of your regular cells. Okay. Now you can say something like, well, there are certain sugars, some saccharides that show up on the surface of regular cells. Yeah, but not with as much frequency as you see on that when they show up on the surface of pathogens. Okay. So you have these things that pathogens have that your cells do not have. So the pattern recognition receptors that are on the surface of your innate immune cells are made to recognize those patterns and get the activation to destroy the pathogen. Okay, so on the surface of bacteria, you find peptidoglycan. You can find lipopolysaccharide. Okay, so this is LPS. This would mainly be gram-negative bacteria, right? We have lipoteichoic acid and gram-positive bacteria. In case of fungi, you have this specific disaccharide that is called mannan, right? So this is specific for fungus, okay? So those are all patterns that can be recognized by pattern recognition receptors, okay? So what kind of PRRs do you have? You have some PRRs that will induce phagocytosis, and you have some other PRRs that are going to induce cell signaling pathways, okay? So the ones that induce phagocytosis, they will bind here to your target pathogen, and they will eat up the pathogen, digest it, and chop it up into pieces. You know, this is what happens when those PRRs are activated. Now, in case of your signaling PRRs, they do get activated. They do not induce phagocytosis, but they induce this cell here to start producing some kind of cytokines or signaling molecules that are either going to go through the inside of the cell and send a signal to really kill the pathogen, or they're going to be exocytosed to send signals to other cells. Okay, so internal signals or exocytosis signals. Okay. All right. So here we're getting close to the end of this whole specific aims for those for this first week, and this idea of how to kill the pathogen. So the first way, as we have seen, is by eating them up. So that is your phagocytosis. So two main cells of the innate immune system, they're very important to do phagocytosis. Those are your neutrophils, and those will do phagocytosis like on stuff that is floating in the blood, or your macrophage. Okay, macrophage is more like tissue resident. And if you're playing the video game thing, so you have the little macrophage that is, doing the job there. Other ways of killing pathogens. You can directly put some, toxic stuff in the pathogen, especially when the pathogen is bigger than the cell. So here, for example, you have killing of tapeworm by certain eosinophils, right? So this is pathogen-directed killing. The third way is by killing the whole cell that gets infected in case your pathogen is inside of the cell. So 3 ways here of killing your cells, okay? And a specific aim 6 on how cells communicate. They can either touch each other or they can send signals to each other. So that's pretty much simple, right? Cell-to-cell contact or molecules. And finally, we get to the ending of the response, right? How do you stop the response? Well, basically, when you get rid of the pathogen, you don't have the stimulus anymore, because the pathogen is really important to be hanging around to keep on stimulating those immune cells to keep on working. If the pathogen is gone, then the immune cell kind of gets bored and died. If it doesn't die, you have a problem of chronic inflammation. So you want the cells to undergo clonal deletion and die, otherwise disease will show up, okay? So basically, this idea of not having your immune system attacking your own cells is what is known by tolerance. That's the word that escaped me before, okay? So peripheral tolerance is teaching your cells to not attack your own cells, right? So in order to really make sure that the immune response ends, besides the cells getting bored and dying, you have these other helpers. They're called T regulatory cells, okay? They really send signals to the activated cells for the cells to be less active. They really put those cells in like antisocial modes, and they really slow them down. So if the cells are not already slowed enough on their own, then Tregs get there and say, all right, guys, there's nothing to fight. There's nobody else to fight. The bad guy is gone. Now let's all, be nice and quiet and either die or be anergic, meaning don't react to anything else. Right? Just float around like a zombie for some time and disappear. Okay, so that's the job of your T regulatory cells. Okay. All right. So this brings us the idea of tolerance, right? So tolerance means that you're not going to attack your own tissues. But then tolerance is a process that is not just happening because you should not attack your tissues. Tolerance has to be maintained. You have to always teach your new cells to become tolerant. Okay, so when you're producing new cells in the bone marrow, they have to undergo this whole developmental stage of learning not to attack your own cells. If anything goes wrong, either in the bone marrow or in the thymus, you're going to have cells that can potentially start attacking your tissues. So something goes wrong in your bone marrow, you're going to have mature B cells that can start producing antibodies against your own tissues. Something goes wrong in the thymus, you're going to have T cells that can start to attack your own cells. Okay, so tolerance really must be maintained, and during the process, you're going to teach your immune cells what is supposed to attack and what is not supposed to. Okay. Then we come to this idea here of immune system going crazy. Okay, all different kinds of things can happen. You can have the immune system that is overreactive and is really attacking stuff that is not supposed to, like for example, pollen, right? So this type of problem you call hypersensitivity. Okay, so you can either have allergies, it can be skin allergies or respiratory allergies, and sometimes it can get even more severe, and you can develop asthma. So these all types of hypersensitivity. Other problem of the immune system is that you can have autoimmune disease. This is a problem when your cells attack tissues that they're not supposed to. So attacking your own tissue is autoimmune disease. So example of this can be multiple sclerosis, right? Or Crohn's disease in the intestines, okay? So this is really example of autoimmune disease. Then on the other hand, you can have problems of not enough action of your immune system. So that is called immunodeficiency, okay? You have two types of immunodeficiency. You can either be born with it, so this would be some defect in the immune system, or you can acquire it later in life. So in relation to primary immunodeficiencies, those are the ones you are born with, and there are many different types of primary immune diseases, immunodeficiencies, okay? In case of secondary immune deficiencies, there is only one example that we know for humans, and that is AIDS. Okay, so this is HIV that causes secondary immunodeficiency. Okay, so when you have an immune system that is not working properly, you can have you can give the opportunity for some pathogens to cause more trouble than they would usually cause. If actually this subdivision here is not correct, I should not put this very close to secondary immunodeficiencies. Okay, so this is like a different idea here. There should be more space here. So an example here of a type of not fully developed immune response you can be seen with thrush, okay? So this is when your immune system is not really containing the yeast. This is what is it? It's some form of yeast that can grow in mucosal tissues, okay? So what happens here, when you don't have proper reaction of your immune system, you can develop these opportunistic infections. This is actually common in nursing babies, in babies that are being breastfed. Okay, so you can have just the skin contact, and you can pass thrush to the baby, but it's easily contained, it's easily taken care of. Okay, but this is a type of response that is not adequate due to the presence of the opportunistic pathogen. Okay. Okay, and this should also be here. All right, so this is a type of practice problem that for you to get familiar with the terms and knowing what they mean. All right, so this, I don't even know if I can put this on iClick or not, but you can practice that on your own. Okay, on exams, I'll not give you questions that are this complicated, like usually, okay? Let me see what is the last one here. All right, special circumstances, the last slide, and I still have like 1 minute to go here. All right, so when you have transplant of tissues, okay, transplant of anything, you know that you're putting something foreign into the body, and if something foreign is not recognized as self, then your immune system is going to attack it, right? So when you do tissue transplants or organ transplants, a big problem that you have is rejection. So usually you give drugs to the recipient, in order to avoid this massive immune rejection. Okay, and when we get to transplants, we're going to see how that happens. Yes.
Because of the drugs, are they more like immune to every other kind of illness?
No, it's actually the opposite. Because of those drugs, they're more susceptible to infections. Yeah, yeah. They're more susceptible to every kind of infection. Yeah. So immunosuppressants make you more vulnerable. So this will be a kind of population that will be at risk and would depend on herd immunity to keep them safe. Yeah. All right. Another special situation that we can see is in case of cancer development. Because we know that our immune system cells are always killing cancer cells, like every day, okay? But sometimes the cancer cell is not killed right away, and it escapes the immune system. Once a cancer cell escapes the detection, it's very difficult for the immune system to go after the cell and kill it. Okay? This is why it's also difficult to develop vaccines against most cancers. There are some vaccines available for some types of cancers, not directly against the cancer, but against viruses that cause cancer development. So those are, still viable, but having vaccines that are able to identify certain cancer cells, that becomes more challenging. Okay, so those are the special circumstances that we see in special topics more at the end of the course. Yes, I know it's a lot of information, but I will put up a quiz for this weekend, and the quiz is going to be due, next week. So,